I think it was 2011 was the last time I contributed to the educational program. I went back and looked at those slides when I was preparing my talk today. It was quite instructive, actually, how much was in those slides that I think we can now take for granted, and also how much the field has grown, and in some quite unexpected ways. And without wanting to, spoil the conclusion of my talk today, I would think the one thing that I really didn't see coming in 2011 was scale, and how important it is that we operate at scale to understand both mechanistic and predictive approaches to brain connectivity. The other thing I noticed when I looked back was I'd included far too many slides in my talk, and I guess what, I've made exactly the same mistake 10 years later. So wisdom doesn't always come with age. I will press on. These are my disclosures. As Klaus mentioned, I worked for GSK half-time for many years. I don't currently, I'm just employed by the University of Cambridge. And what I thought might be interesting for people is to take a fairly sort of high-level view about, what do I mean by mechanistic studies of brain connectivity, and at least for the purpose of the talk, I'm going to understand that phrase to refer to studies that mostly have a kind of analogous logic, or use some kind of correlational analysis to try and build a bridge between some kind of MRI connectivity metric on the one hand and some biological mechanism on the other. And often that endeavor will involve a kind of orthogonal axis of translation between humans and non-humans. And the MRI connectivity metrics could be many and various. There could be multi-structural MRI parameters, MRI gradients, graph metrics of network topology, and the biological mechanisms equally diverse. Neurons, synapses, axons, it's not surprising that we think of those as biological mechanisms of brain connectivity. Also cortical layers, cytoarchitectonics, genes and transcripts you've already heard a bit about. And I will touch on the immune system, as Klaus indicated. That is, I think, an interesting and emerging area. So the general logic is exemplified by the efforts that have been made to link MRI markers of myelination to underlying neurobiological processes at a cellular level. And I think that is an anchor point for mechanistic studies of human brain connectivity, but not the only one. Predictive studies, on the other hand, I will characterize as having a slightly more reductionist logic and or attempting a more causal analysis. So the arrows in all of these diagrams that I'm about to show you only have one arrowhead, implying a causal direction, and that could be in the context of development, where you're looking at some MRI connectivity metric at time t, predicting the same metric at time t plus 1. They could be causal in the sense that we're looking at the ultimate cause of biological variation, that is sequence variation in DNA, or possibly epigenetic modification of the genome, and how those factors, which one might regard as respectively genetic and environmental, or genetic bioenvironmental factors, play out in terms of some MRI connectivity metric. And I'll say something briefly also about computational models, generative models, such as the ones that Alex has just discussed, where you start with a model and try and simulate the connectome. And I'll also say something very briefly about the situation where you start with a connectome and use that to inform a neuromorphic model of cognition. So let me get cracking on mechanistic studies. And as I said, I'm going to go fairly high level and quite rapidly, but just to try and sort of survey the territory. One of the big things that has come along, at least in my understanding of the field, that I didn't see so clearly back in 2011, is the importance of multi-structural MRI. So being able to, at voxel resolutions, more or less simultaneously acquire multiple parameters. And from those multiple parameters, then estimate quite a wide range of structural phenotypes, which could be macrostructural or microstructural, and people may be already familiar with this distinction, but macrostructural phenotypes I understand to be the kind of classical candidates, cortical thickness, cortical surface area, and volume, all of which are estimated by data from multiple regionally parcellated voxels. And then in contrast, you have the microstructural phenotypes, magnetization transfer, proton density, neurite density index, etc., all of which can be estimated by data from a single voxel, though in practice they're often regionally averaged. And the picture there just shows one sequence for multi-parameter mapping produced by Nick Weiskopf and colleagues. And in the bottom row, you can see that that has quite low between site variability at 3T, which I think is important in terms of scaling up over multi-site studies. As I've already said, I think the importance of intra-cortical MRI as a marker of myelination is quite important. To me, it's a key anchor really in understanding brain MRI connectivity. And the picture on the left is, I'm sure, familiar to many people. This is from the Glasser and Van Essen paper in Journal of Neuroscience 10 years ago that really highlighted the credibility of the T1-T2 ratio as an indicator of intracortical myelination. At 3 Tesla, these data were required. On the right are some results from Bob Turner's group at the Max Planck Institute in Leipzig, where they used 7T to map T1 alone, and in ex vivo brains, where they could make very precise measurements of the T1 gradient through cortex, they were able to align those results quite precisely with histological preparations from the same brain. So that's, I think, a nice example both of integrating histological and imaging data to find cellular substrates that make sense of the imaging data, but also actually of using the higher field strength that we have at 7T for a very good purpose, which is to essentially push the near laminar resolution of cortical metrics that we can measure in vivo. Now, with those kind of data, one of the things that can be done, and this is an example of my first introduction to this. This was work by Kirsty Whitaker and Petra Vertes a few years ago now. And what you can see on the left here is a sort of cortical gyrus and a transect running through it. And you can probably just see in the background here, the slightly pixelated background, those are NT measurements at individual voxels. And by interpolating across voxels within a region of interest, you get this sort of subvoxel resolution of the MT signal increasing as you go from the peel surface down to the gray-white matter boundary. It looks as if it might represent cortical myelination, certainly when you line it up against some of these classical histological data, and this is from the votes from about the turn of the 19th and 20th century. I think you can see quite clearly there in their histological preparation, denser myelination of the deeper cortical layers, which is at least by eye consistent with this increasing MT signal as you go from peel down to the gray-white matter interface. That was in 2016, and then a few years later, we've got a sort of generalization and extension of that approach. This is work by Casey Pequola and Richard Bethlehem. Again, post-mortem being compared to in vivo MT, and here you've got profiles of intensity from myelin-stained cortical slices that are being compared with the in vivo profiles of magnetization transfer estimated through the cortical thickness. And I think you can see, again by eye, that there's some correspondence between the forms of the intensity profiles in histology versus MRI across the different cortical areas, V1, V2, and so on. Here on the right, you're seeing some summaries of the statistical properties of these profiles. So if you consider these profiles as probability distributions and look at their mean, their skewness, and some of their other moments, what you can see here is that the different cortical areas have the same rank order, at least almost always have the same rank order in terms of their mean and their skewness, again suggesting that what we're measuring with MT is at least statistically analogous to what we can measure histologically in a range of different cortical areas. Here's another example, and I guess again this picks up on the big brain, which Alex just mentioned, and of course is work that's been driven in part by the collaboration between Montreal and Ulick. This is the 40 micrometer resolution image of a cortical gyrus showing the very, very careful delineation of cortical layers. And this as a resource has been used to estimate cortical thickness histologically with great precision, and then cross-validate the cortical thickness measurements that we have histologically with what we can find in the literature and what we can measure using MRI as obviously an in vivo marker of cortical thickness. So these are all examples of looking in detail at cortical structure, trying to think about cortical lamination, trying to validate it using histological measurements we have available. These pictures are showing a bit of a story that I'll come back to, which is the importance of development in thinking about the brain organization in adult life. These are from a study in about 300 healthy young people aged 14 to 24 years old. We call it the N-span study. And here you can see the magnetization transfer at 14 years with the more putatively myelinated regions shown in red. And here on the right, you can see the rate of change in myelination over the course of adolescence, with most of the action happening in association cortical areas. And when we first saw this map, one of the things we asked ourselves was, well, how can we validate that biologically beyond the kind of histological correspondence that I've just described? And one innovation that came out of it, at least from my point of view, was the concept of combining those imaging phenotypes with what was then recently available human atlas data on brain gene expression provided by the Allen Institute, and combining those high-dimensional phenotypes, the gene expression and the imaging, with a multivariate technique like partial least squares, which incidentally Randy was the first to introduce into our field even longer ago than 2002. I think it was 1996 was the first PLS paper from McIntosh and Bookstein in the imaging literature. Anyway, it's turned out to be an incredibly useful multivariate tool for a number of different reasons, but in this context, what it gives you is a ranked list of genes across the whole genome that are weighted so that their combined expression profile is as spatially co-located as possible with the imaging phenotype of interest, which is great, but you've then got a ranked gene list to make sense of. And there are, of course, a number of bioinformatic tools that you can use to do that. And this is the result that we came up with back in 2016, showing that this pattern, this phenotype of adolescent increase in magnetization transfer, putatively intra-cortical myelination during adolescence, was co-located with a gene expression profile that was enriched for a number of synaptic and oligodendroglial genes. It was also actually enriched for genes related to schizophrenia, a point that I'll come back to later. And Alex and others have really sort of advanced this approach of using transcriptional data as a kind of point of reference to make sense of many different imaging phenotypes. So intracortical myelination, I like very much. I like the fact that it's something that MRI is good at measuring. An MRI machine is basically a device for telling the difference between fat and water. In the brain, most of the fat is myelin. So you're asking MRI to do something it likes doing, if I can use that expression. And of course, the fat measurement corresponds to something that's of keen biological interest to us when we're thinking about connectivity. So that's why I like it as an anchor. But it's obviously not the only biological process that's important in terms of brain connectivity. And another one that we often talk about, particularly in relation to development, is synaptic pruning. And this little graphic I just took out of a review by Beth Stevens and colleagues. Beth Stevens has been one of the leaders really in thinking about some of the neuroimmunological mechanisms that might be at play during development, and particularly she's highlighted the role of the microglia, which are the innate immune cells of the brain. They're a bit like macrophages, but they sit in the brain rather than other tissues. And they're phagocytic cells, which in terms of response to infection actually eat bacteria and destroy them digestively. And it turns out that in relation to the development and maintenance of the connectome, these phagocytic microglial cells may be quite important in actually eating synaptic terminals and pruning them in that very literal sense. I think that's just an example of how important it may turn out to be, the interactions between the immune system and the nervous system in terms of the development of connectivity. I'll just show you an example of a much sort of simpler sort of neuroimmune study that Manfred Kitt-Spickler led on and we published last year. This uses the microstructural metric of proton density that I've already alluded to briefly. Proton density is essentially a measure of the amount of the density of protons. So to a first approximation, it's a measure of the amount of water in a voxel. And this is a map showing where in the brain proton density was correlated with plasma levels of CRP, C-reactive protein, one of the body's most widely measured markers of innate immune system activation. And here you can see in particular it's this posterior cingulate precuneus region where there is a significant positive scaling relationship, so higher levels of CRP going with higher levels of proton density. That's kind of interesting because if you look in the literature around PD in well, particularly neurological disorders, stroke, multiple sclerosis, hepatic encephalopathy, PD and increased PD are quite often interpreted as measures of extracellular edema. It's perhaps too simple to be true, but I quite like the idea that part of the measurable brain response to systemic inflammation may be a degree of local swelling in some brain areas that we can pick up with this microstructural marker. It's also notable that this anatomy in the posterior cingulate coincides anatomically with some of the areas of the brain that we see functionally disrupted in depression. So this is a disconnectivity map of cases of depression compared to control, showing reduced connectivity in default mode network nodes. And Manfred used the technique of mediation analysis to try and join these things up a little bit. So here we can see that for at least some connections between the posterior cingulate and the medial prefrontal cortex, the increase in CRP, the increase in peripheral inflammation is explaining the variance in proton density in the posterior cingulate, which in turn is explaining the disrupted connectivity, functional connectivity between posterior cingulate and medial prefrontal cortex. So this is an example of how, using a very simple peripheral inflammatory marker, we're beginning to understand how it might relate to microstructural and functional connectivity changes in circuits that are relevant to depression. And at that point, I would say we reach a bifurcation point, as is traditional during a brain connectivity workshop talk. And we can take one of two roads. We can either zoom in and ask ourselves, well, what are the biophysical mechanisms driving variation in these voxel-wise parameters in more detail? You know, does increased MT indicate increased myelin per neuron or increased density of myelinated neurons, for example? Is the neurite density index a measure of synaptic density, dendritic arborization, axonal myelination, or all of the above? And those are interesting questions, but they're sort of very detailed. Alternatively, we can zoom out and say, well, okay, well, what are the network properties of interregional covariation between some of these multi-MRI parameters? And it might not surprise you, I'm going to zoom out. I'm going to leave some of those more detailed questions on the table, and I'm going to start talking more in the language of networks and connectivity at a systems level. I'm not going to walk through this in detail. This is this one picture probably summarizes most of the slides that I presented back in 2011. The idea that you can, from MRI data, get down to graphs and look at their topological properties in various different ways. Now, one way of doing that is called morphometric similarity mapping, and this is a technique that was first written up, as far as I know, by Jacob Seidlitz and colleagues a couple of years ago. And what it involves is starting again with this multimodal MRI parameter set, parcellating those data, and from the multiple parameters available at each region, constructing a feature vector. So this has 10 elements in it, 10 different things we know about each region as a result of analyzing these multi-parameter data. And the morphometric similarity is very, very simple. It's just the correlation between two regions in terms of their multi-parameter feature vectors. And you repeat that correlational analysis for every possible pair of regions in the template, and you end up with a matrix. You can binarize it, you can represent it as a network, either in anatomical space or topologically. We can see that in a little bit more detail here. This is a map of nodal similarity. In fact, this is the signed weighted degree of the morphometric similarity network. And you can represent it topologically. You can show that it has a modular organization. It contains hubs. It contains a rich club. It has that suite of features that we've seen in many different brain systems. So it looks like a brain network, and it's tempting to say, well, let's call it a connectome. But is it really a connectome? In other words, how confident can we be that morphometric similarity measured in this way actually tells us something about the axonal connectivity between areas? Well, one reason that we feel it might have something to do with connectomics is, again, because of basic histological work. And this is a study from Klaus Hilgetag's lab. This is a flat map of macaque cortex highlighting neuronal density. And then on the bottom, you can see the density ratio. That is to say, effectively in log space, the difference in neuronal density between cortical areas. So these, this bar here with a low log ratio density indicates that there are a number of similarities, at least in terms of neuronal density between two cortical areas, and the areas or pairs of areas for which that is true tend to have a more higher probability of an axonal connection between them. So the point being that if you look histologically in these and other examples, you can see evidence that cytoarchitectonic similarity tends to predict myeloarchitectonic connectivity. And that's one reason that we think that morphometric similarity might have something to do with axonal connectivity. Now, we tested that hypothesis in the imaging data. So these are results from a morphometric similarity network, a structural covariance network, and a DWI-based tractography network, all in the same data. And we reasoned that if it's true that cytoarchitectonically similar areas are more axonally connected, then we would prefer our MRI-based measure of the connectome to include a large proportion of connections between the cytoarchitectonically similar areas. And that indeed is what you see. This is the density of connectomes at various proportions of the total number of possible connections. So these are quite sparse networks from 1 to 5%. But you can see over that range, the morphometric similarity network includes a higher proportion of edges that belong to the same cytoarchitectonic area. I think you can see that also in the plots here where the nodes are colored according to the cytoarchitectonic area. So that's encouraging. It helps us believe that the morphometric similarity we can measure with MRI is telling us something about axonal connectivity. But as you all know, I'm sure, the gold standard for inter-areal anatomical connectivity is tract-tracing, which is not a technique we can use in humans, but we can, of course, in animals. This is a figure from a paper that Mika Rubinoff and colleagues did using data again from the Allen Institute, but this is with the mouse brain, looking at what happens when you inject a tracer in a particular source area, wait a few days, sacrifice the animal. You can see that tracer has propagated antegrade to its target area. And from those measurements, you can very precisely work out the connection density. You can also work out the axonal wiring cost. You can explore the topology of this network. It has, for example, a modular organization. And you can also make similar measurements in a monkey. And this is from the Seidlitz et al. Paper. This is tract tracing data from the macaque being compared to morphometric similarity data derived from imaging data in the macaque. So this is imaging, this is tract tracing, and this is the correlation between them. It's not the tidiest scatter plot in the world, but it is a significantly statistically significant association consistent with the idea that the tract tracing measurements of anatomical connectivity are related to the MRI measurements of morphometric similarity. So those are a few vignettes of sort of what I might call mechanistic studies, trying to make sense of MRI metrics. I'm now going to just rattle through, because I'm conscious of time, a few examples of predictive studies. And with apologies, I am going to have to go quite fast, but the slides will be available if anybody wants to look into any of this in a bit more detail. So the first thing I would say about development is I genuinely think we are at a bit of a sort of on the cusp of a slightly different way of thinking about brain development, which again follows from scale. So this is data that was published early this year, based on 130,000 MRI scans from before birth till about 100 years of age, plotted on a log scale here. Just to remind us how important the first period of gestation and the first 5 or 6 years of postnatal life really are in terms of changes in a number of MRI phenotypes, but cortical thickness, volume, surface area, gray matter volume, white matter volume, and so on. All of these markers are very active and in fact often have significant milestones in the first 2 decades of life, coinciding, of course, with the period of highest incidence of many psychiatric disorders. This is just to show you those data in a little bit more granularity. So you can see here the 130,000 scans. You can see the trajectories that have been estimated from them using a technique called generalized additive modeling of locations, scale, and shape. You can measure the trajectories. You can measure the between subject variance in those phenotypes over time. You can take the derivatives and you can look at the rates of growth. And again, you're seeing that the maximum rates of growth for surface area and volume are in the first year of postnatal life, and cortical thickness actually peaks about a third of a year before birth. I think Alex mentioned this concept of regional heterochronicity, which you can see again in these data. So the growth curves here for total gray matter volume show some regional variation around the point of the global mean, with for example, areas of the insula maturing later than primary sensory or motor cortex. This is just a little snapshot of the methodology behind all of that. So measuring the phenotypes, taking into account some of the demographic variables, and then using this GAMLS-S modeling technique, particularly to adjust for site-specific differences. So one of the big roadblocks to scaling MRI has been this question of how do you make it work across multiple sites? How do you harmonize data that might have come from different scanners and so on? And this is a statistical technique that does that kind of batch correction very elegantly by essentially giving each study its own distributional offset in terms of mean variance and skewness. And then from that, you can estimate centile scores, which tell you roughly, well, not roughly, but they tell you precisely for each individual in a new study, where does that individual sit with respect to the normative trend. And the reason I think this is important is not just because of that study, but almost simultaneously, a Dutch group published a very similar method in Nature Protocols, again highlighting that this is now something that will allow us to think about how we describe people as abnormal or atypical in terms of their brain development. This is a really rather different way of doing that by comparison to these normative growth charts and using statistical techniques like gamma LSS to think about individual atypicality. I see two big gaps still to fill. One is around perinatal life, which is so important, as I've just shown you in those data. And this is a picture from a review by Dubois et al., which is a nice introduction to some of the methodological challenges in perinatal imaging. This, you can see, is an abnormally shaped head. It's abnormal in the sense that this child has just been born, so that the skull is still deformed by passage through the mother's pelvis. You can also see the gray-white contrast is rather opaque, and there are also a number of artifactual challenges in perinatal imaging. But I think that's going to be a really important area for us to see more data as a field. The other point that's missing is diversity. So this is a map showing where those 130,000 scans came from. And you can say, well, we've got some contributions from all over the world, but you can also see at a glance that actually that's mostly from North America and Western Europe. And if you dig a little bit deeper, most of those data sets are from relatively affluent demographic sectors in those countries. So we have far from complete global coverage of brain development, both geographically and socioeconomically at the moment. I think that's going to be something important for us to think about in the future. One aspect of individual differences and demographic effects that we can already look at is sex. I sort of slipped over this a little bit when I was talking about the brain charts, but all of those were sex stratified for men and women separately. This slide that I'm going to talk you through, it's a bit of a build, shows how you can begin to sort of explore sex differences in development of functional connectivity. So we start here with a number of repeated observations of functional connectivity at the age of 14 and of the linear rate of change in functional connectivity over the 14 to 26-year-old range. And we, František Vassar and colleagues combined these data to estimate something they call the maturational index and showed that in some brain areas, we call them conservative, you have a high functional connectivity at 14, and there's further increase in functional connectivity during adolescence. In other areas, we call those disruptive, you have low connectivity at the age of 14, but it becomes more strongly connected over the course of adolescence. So this seems to be an indicator of new functional connections emerging during the course of adolescence. And Lena Dorfschmidt showed that there were some quite interesting sex differences in terms of that pattern. Particularly females showed a more disruptive pattern of connectivity development in these blue areas here. So the females are shown in red in this scatter plot, low functional connectivity at 14, but becoming more connected during adolescence, whereas males stay about the same, which was interesting to us because there are a lot of sex differences in risk for mental health during adolescence. Young women in particular have a much higher incidence of depression in the 14, 15 to 16-year-old range. We wondered, is this phenotype relevant to depression. So you can do a kind of neurosynth search and find psychological functions that are associated with those regions, and they include a number of reward and motivational processes, which is kind of consistent. We correlated this phenotype with the case control difference in connectivity in major depressive disorder, and indeed it was correlated. So this developmentally disruptive pattern is anatomically consistent with brain systems that we know are disorganized in depression. And then finally, we could do the transcriptional integration with partial least squares and show that the gene expression profile that was most co-located with this disruptive development phenotype was enriched for genes in the X chromosome, genes that were developmentally expressed during late fetal life, and genes that had been previously associated with risk for major depressive disorder. So, this to me is kind of interesting, beginning to get to grips with sex differences in functional connectivity development, how that might be related to sex differences in the emergence of mental health disorders. This is beginning to poke our toe in genetics. And this slide on the top is summarizing essentially a sort of association analysis. You take a number of different imaging markers, number of different imaging phenotypes on the one hand, and also from published GWAS studies, in this case of schizophrenia, you can construct polygenic scores of risk and then measure the association between polygenic risk for schizophrenia and multiple structural MRI phenotypes. And the key result in this work by Eva-Marie Stauffer was that genetic risk for schizophrenia in particular was associated with reduced neurite density index and a number of other phenotypes of cortical myelination in adult brains from the UK Biobank cohort in these locations. Now that's interesting to me, at least, because besides being highly heritable, schizophrenia is neurodevelopmental, and the fact that the risk genes for schizophrenia are associated with these variations in a microstructural MRI marker of connectivity is intriguing. We can pursue that kind of link between the genetics of schizophrenia and network organization or large-scale brain connectivity in a slightly different way. And this is recent work from Sarah Morgan and colleagues, starting with a morphometric similarity network in healthy controls, then looking at the case control difference between patients with schizophrenia and healthy controls in terms of morphometric similarity. So these blue areas are where there is reduced similarity in the cases. And as you can probably see by eye, these two maps are strongly negatively correlated with each other. So the areas that have reduced similarity in schizophrenia are those regions precisely that have the highest similarity, putatively the hubs of the morphometric similarity network in the controls. And then we can begin to explore what that means, again, using this technique of partial least squares to identify a weighted gene expression map that is co-located with the case control difference in morphometric similarity. And here we're looking to make sense of that result, not by the kind of enrichment analysis that I described on a previous slide, but using a protein-protein interaction. So this is another kind of network, but it's a network of interacting proteins coded by the list of genes co-located in terms of their expression with the case control difference in schizophrenia connectivity. And this protein-protein interaction network shows a lot of interactions between proteins, which are probably illegible on this slide, but include, for example, the dopamine D4 and D5 receptors and a number of other proteins that have been implicated either in schizophrenia or its treatment. I've got, as I said at the start, too many slides. but I'm going to just keep running through them just quite quickly because some of this stuff I think is rather wonderful. This is a paper from Oxford, of all places. They started with, again, multi-parameter MRI data, and then asked themselves the interesting question: does the multi-structural profile of callosal tracts have anything to do with the strength of functional connectivity between homotopic cortical areas? So here you can see they've got the skeletal analysis of a callosal tract, and then they've basically measured all the microstructural parameters in the white matter that emerge from the callosum and related that to homotopic functional connectivity. And shown here that you can do a reasonably good job. I mean, so it can accounts for about 12% or so of the variance in homotopic functional connectivity can be accounted for by the microstructure of the callosal tracts. What I think is also important is to highlight they've done this in a training cohort, they've done it in a replication cohort, both quite substantial, several thousand individuals, quite robust. And then to get a genetic hook on this, they didn't do the usual thing with the Allen Brain Atlas. They actually did a GWAS. So they did their own GWAS, and they discovered that there were a number of significant hits. In particular, here on chromosome 14, they found some genes that were important in predicting this microstructural functional coupling phenotype. Okay, so it does get a bit intricate what they've done, but it is also quite a powerful example of how you can run all the way through from multimodal MRI structural function coupling to GWAS and identifying specific variants that might cause phenotypic changes. How long should I realistically aim to keep talking for, Klaus? Do you want me to stop now? Or?
You're at -2 minutes, but we're happy to have you for another 5 minutes.
Another 5. Okay, well, that's very generous of you. So I'll just keep going then. I talked about the immune system a couple of times, and I would recommend, if you're interested in that, you're interested in thinking about how it might relate to development of brain circuits. These papers by Nusslock and colleagues are well worth reading. They have a neuroimmune network theory, which basically supposes that early life stress can cause changes in the immune system, which in turn can cause long-term changes in network development. And they've also sort of gone some way to empirically validate this in studies of African-American youth. I highlight that point in relation to what I said earlier, which is that neuroimaging research has tended to be a little bit sort of indifferent to socioeconomics. And the corollary of that is that actually a lot of more disadvantaged communities have actually been sort of somewhat excluded from imaging research. I think it's commendable that Nusslock and colleagues have gone out of their way to recruit traditionally difficult to access groups, and shown here that connectivity in this emotion regulation network is associated with inflammatory markers. When you think about early life stress causing long-term network changes, you might want to ask yourself, well, how could that be? What would the molecular mechanism of that be? And I think epigenetics is going to be part of the answer. We've tended to kind of, or I have tended to shy away from epigenetics on the grounds that you need tissue-specific measurements, and the brain is not the most accessible tissue. But these graphics from work by Elizabeth Binder, who's been a leader in this field, highlighting that there's a growing awareness that actually you can measure epigenetic changes in peripheral blood samples that might be relevant to epigenetic modification in the brain. And this paper by Chen and colleagues, I think is really quite remarkable. So they did first of all, did a whole blood methylomic profile in patients with schizophrenia and healthy controls, and found that there was a methylation profile, which was somewhat predictive of diagnosis. Then they did sort of enrichment analysis on the methylation marks and found that they were concentrated in pathways that are important for the immune system and synaptic processes. And then they showed that the methylation, what they call the polymethylation score, so it's a bit equivalent to the polygenic risk score that I mentioned earlier, that this is methylation aggregated over the whole genome, was correlated with connectivity of the hippocampus, or connectivity between the hippocampus and the dorsolateral prefrontal cortex. I think that might be the first study that's looked at whole methylation in relation to brain connectivity, but I don't think it's going to be the last. And now I've got into my whole usual thing about Cajal and conservation laws. So I'm coming to the end. You'll be delighted to hear. I was going to talk about computational stuff and highlight that this idea of Ramon y Cajal's, that a lot of variation in neuronal morphology is due to laws of conservation for time, space, and material. He inferred that from this famous Shepherd's crook neuron. There's a Shepherd's crook, and that's what he was seeing in the neuron. He wondered why the axon emerged here rather than close to the cell body, and he reasoned that it was to do with a trade-off between conserving material, that is axonal length, versus time. In other words, minimizing the amount of time it took for electrical impulses to travel from the dendrites out of the axon into the rest of the nervous system. And this is a trade-off that I think you've probably heard a bit about already from Alex and others. To my mind, this paper again by Kaiser and Hilgertag is one of the first to really show such a trade-off between, in this case, wiring length and topological integration. In the macaque and also the C. elegans, you can make the wiring cost less, but you do so at the expense of path length and other measures of topological integration. And that encouraged Olaf and I and others to think about economical principles of brain network organization as quite likely fundamental. And you can write those laws down formally, and you've just heard from Alex something about this equation, which stipulates that the probability of connection is a product of the distance and the topological relationship between nodes. This we first explored with Petra Vertes in fMRI data and has since been replicated by Rick Betzel in DTI. And also more recently, Dana Karka and colleagues have looked in some detail at these generative models and shown how tightly constrained the parameters must be, but also that we can begin to put some biological flesh on this, again using, in this case, the transcriptional analysis based on Allen Brain Atlas. You can show that there are different gene sets enriched for the two parameters, the distance penalty and the weighting on topological homophily. And very finally, I will talk about this paper, which I also think is an excellent paper from the Misic group. Basically, they've built a reservoir computing model which has a connectomic topology and shown that the memory capacity of this system depends quite sensitively on both it being a connectome and not some kind of randomly permuted network. It depends on the topology, and it also depends on the coupling strength being positioned close to the point of criticality in these dynamics. And I think that is a very interesting future direction for the field. Again, this neuromorphic computing and knowing what we increasingly know about the connectome to simulate cognitive functions. So I've got to the end in a bit of a sort of scramble, but basically I've tried to highlight a couple of areas where I think mechanistic work has moved ahead, at least in my mind, quite convincingly. And I've pointed to some of the sort of more predictive approaches that I think might be important in future. I put that map up in 2011 to signify that we might not know everything, but we're beginning to know something. And I think that's truer now than it was then. So the map might not be complete yet, but I think we are definitely moving as a field in the right direction. And finally, I'd just like to thank those people that I've had the pleasure and privilege of working with on some of the studies I've shown you, and to reiterate Alex's shameless plug for the fundamentals of brain network analysis.